Okay, let's unpack this. Today we're taking a deep dive into a compound you might have heard mentioned quite a bit lately, actually. TUDCA.
Tauroursodeoxycholic acid. Yeah, thank goodness for the abbreviation.
Absolutely, TUDCA it is. There's definitely some buzz building around it, especially in like biohacking circles. People are talking about it for liver support, brain health, even boosting mitochondria.
And, well, like a lot of these interesting molecules, there's a bit of a mix, isn't there? Some solid science and then maybe some stuff that's more theoretical promise at this point.
Exactly. So our mission here for this deep dive is pretty clear. We're going to lean heavily on the source material you've shared,
Yeah.
this evidence-based guide, to really understand what TUDCA is and dig into what the science actually says based on that evidence, trying to separate, the robust findings from the more speculative ideas.
Sounds like a good plan. So maybe start at the beginning. What is TUDCA, fundamentally, biochemically speaking?
Right. So the source describes it as a taurine-conjugated bile acid. Essentially, it's a type of bile acid your body makes naturally.
Okay.
It's formed in the liver, combines something called ursodeoxycholic acid, that's UDCA, with the amino acid taurine. But we only make small amounts ourselves.
And it's not exactly new, right? It has a history.
Yeah.
Our source mentions clinical use for a while now, particularly in Asia and parts of Europe.
Yeah, that's right.
For liver stuff mainly.
Primarily, yes. For specific liver diseases, things involving cholestasis, that's basically when bile flow gets blocked or slowed down.
Right.
But the source highlights something really key. It's not just about digestion. TDCA is known for protecting cells when they're stressed. It's described as a cytoprotective molecule.
Okay, cytoprotective. So it helps cells survive stress. That's interesting. How does it do that? That's where the biology must get pretty cool.
It really does. And one of the best studied ways, according to this guide, is its effect on something called the endoplasmic reticulum, the ER.
Ah, the ER. The
And-
cell's protein folding factory, essentially.
Exactly. Like the assembly line for proteins. But, when a cell gets stressed out, maybe from inflammation, poor metabolic health, toxins, that kind of thing.
The assembly line gets gummed up.
Pretty much. Proteins start misfolding, and that triggers this alarm system called the unfolded protein response, or UPR.
Okay, so stress leads to bad proteins leads to an alarm, and TUDCA.
It steps in. It acts like a chemical chaperone.
A chaperone, like helping proteins fold the right way.
Precisely. It helps smooth out the folding process, clears the jam, and dials down that ER stress response.
Okay, that makes sense. But you mentioned earlier there's more to it, something connecting to mitochondria.
Yes. This is where I think it gets really fascinating according to the source. The ER isn't isolated. It's physically and functionally connected to the mitochondria, the cell's power plants.
How are they connected?
Through these structures called mitochondria-associated membranes, or MEMS. The source describes them as like communication hubs, critical for things like calcium signals, lipid transfer between the two.
Okay, so they're talking to each other constantly via these MAMs.
Right. So think about it. If the ER, the protein factory, is under major stress.
It's going to affect its neighbor, the mitochondria, through those connections.
Exactly. That ER stress can destabilize the mitochondria. It messes with their ability to produce energy, ATP, and makes the whole cell much more vulnerable.
Ah, I see. So TUDCA, by helping the ER,
By calming down that ER stress.
it indirectly helps keep the mitochondria stable and functioning better through preserving that ER-mito link.
That's the idea presented. And the source notes this is potentially a really big deal in metabolic diseases and also neurodegenerative conditions because both ER stress and mitochondrial problems are common themes there.
Okay, so it's not just one specific action, it's about supporting this fundamental cellular stress response pathway. That ER-mito connection seems key.
It really ties it together. It explains why it might pop up in relation to seemingly different things like liver, metabolism, and brain health.
Right. Fascinating mechanism. So let's pivot then. What does the research actually show for these different uses? Based on the guide, where's the evidence strongest?
Okay, yeah. The source is pretty clear here. Liver support. That's where the evidence is described as well established. It's actually led to clinical use.
Clinical use meaning prescription.
Yes. It's an approved treatment for certain cholestatic liver disorders, like primary biliary cholangitis. The guide says it helps improve bile flow, reduces inflammation, and promotes liver cell survival in those cases.
So for those specific diagnosed conditions under a doctor's care, it's a recognized therapy.
Correct. Now, what about something like, say, NAFLD, non-alcoholic fatty liver disease?
The source suggests it's showing promise there, but the human trials are noted as being smaller and more research is ongoing.
Okay.
It does mention a typical dose used in some NAFLD studies around 1,000 milligrams per day.
Got it. Strong for specific cholestasis, promising but needs more work for NAFLD. Okay, let's move out of the liver. What about insulin sensitivity? That gets talked about a lot.
Oh, it does. Here, the source describes the evidence as promising, but with a really important caveat. It seems specific to certain people.
Meaning?
Well, it points to a small human study. This study involved individuals who were obese and insulin resistant.
Right.
And in that group, taking TUDCA for 4 weeks did improve insulin sensitivity pretty significantly by about 30%.
Wow, 30% is quite a jump for that specific population.
It is. But, and this is crucial, the source makes a point of saying this benefit hasn't been shown in people who are already metabolically healthy.
Ah, okay. So it might help improve existing insulin resistance, but it's not necessarily something for healthy individuals looking for an edge.
That seems to be the takeaway from the evidence presented here. And the dose in that particular trial was higher, by the way, around 7,750 milligrams a day.
Good to note. Higher dose, specific population.
Okay.
What about that core mechanism we discussed, mitochondrial protection? Does the research back that up in humans?
Well, the mechanism makes sense, right? It's theoretically plausible. But the source classifies the direct evidence for mitochondrial benefits mostly as preclinical.
So animal studies, cell studies.
Yeah, exactly. Those studies show some positive signs, things like reducing how permeable the mitochondrial membranes are, boosting ATP energy production, lowering oxidative stress.
So the lab work fits the theory.
It does, but we're still waiting on solid human trial data to confirm those effects directly in people. So the dose for that application, the source just lists it as experimental.
Experimental, okay. How about neurological support? You often see things that affect the ER and mitochondria tested for brain conditions.
You do. And look, there have been some encouraging results with TUTA in animal models. Studies looking at Parkinson's, Alzheimer's, even stroke models have shown some promise.
But I sense a but coming.
Yeah, there is one. The source brings up a really important counterpoint. A phase 2 clinical trial in humans with ALS, that's motor neuron disease.
Right.
That trial unfortunately failed to show a significant benefit from TTCA.
Oof. That's a significant finding, too. It really highlights that jump from animal models to human results isn't always straightforward, even when the mechanism seems like it should work.
Exactly. So for neurological support, the source essentially lands on mixed evidence. You've got promising preclinical stuff, but it's bumping up against inconsistent, or in the case of ALS, negative results in early human trials so far. Dose here is also marked experimental.
A mixed bag, then. Okay, one last area the source touches on, gut health and the microbiome. What's the story there?
This is painted as more of an emerging research area. The source talks about some plausible, indirect ways TDCA might play a role.
Indirectly how?
Well, 2 main ideas. First, bile acids themselves, despite being in the gut, can act as natural antimicrobials, so they could potentially influence which bacteria thrive there, kind of shaping the microbiome balance.
Okay, that makes inherent sense. They're flowing through there anyway.
Right. And second, there's signaling. Bile acids can interact with specific receptors on gut cells. The source mentions names like FXR and TGR5. And activating these receptors can influence the release of gut hormones, like GLP-1, for example.
Ah, GLP-1 is involved in metabolism and insulin signaling, right?
Exactly. So the idea is, through these signaling pathways, bile acids like PUDCA could indirectly affect metabolism, maybe inflammation, insulin response, all originating from the gut.
So potential effects through tweaking bacteria and through cell signaling in the gut lining.
That's the hypothesis being investigated, but the source is careful to state this is still very much an active area of research. We don't have definitive answers yet. And the dose for this specific potential use, listed as unknown.
Unknown. Okay, well, that covers the major areas of research pretty thoroughly. Thanks for walking through that evidence based on the source. Now let's get practical. If someone is considering TUDCA, maybe as a supplement, what does the source say about dosing, safety, that kind of thing?
Sure. So looking at the human studies mentioned in the guide, the doses used have varied quite a bit. Generally, the range seems to be from about 250 milligrams up to that 1,750 milligrams per day we saw in the insulin study.
And often split up.
Yeah, frequently taken in divided doses through the day. And as we just discussed, the specific dose often depended on what they were actually studying, liver support versus insulin sensitivity, etc.
Makes sense. What about safety? Any major side effects to be aware of?
Generally, it seems to be considered well tolerated according to the source. The most common things reported are some mild gastrointestinal issues, maybe some bloating, or occasional diarrhea.
Anything help with that?
Taking it with food seems to be the usual recommendation to minimize that possibility.
Okay. Now, this next point from the source feels really critical, especially given the buzz online. The difference between supplement TUDCA and pharmaceutical TUDCA.
Yes, absolutely crucial. Cannot emphasize this enough. The TUDCA used by doctors for those specific liver conditions, like PBC, that's a prescription drug. It's highly regulated, tested for purity and potency, and used under medical guidance.
Whereas dietary supplements.
They're just not held to the same standards. Not even close. Quality, purity, even the actual amount in the capsule can vary wildly. It's a different ballgame.
So if someone is navigating the supplement market for TUDCA, what advice does the source offer to, well, reduce the risk?
It gives some pretty sensible guidance. First, look for brands that actually get their products tested by independent third-party labs. That gives you at least some assurance about purity and dosage accuracy.
Good tip. What else?
Be really skeptical of brands making wild health claims. If it sounds too good to be true, it probably is. And importantly, try to stick to the dosing ranges that have actually been studied for the specific reason you're interested in it, referencing the doses we talked about from the guide, rather than just assuming more is always better.
And the absolute bottom line. This isn't something to use instead of treatments your doctor has prescribed for serious conditions, right?
Absolutely not. Full stop. Never replace prescribed medical care with a supplement.
Okay, so let's try and synthesize all this. Based purely on the evidence laid out in this source material, what's the final verdict on TUDCA for you, the listener?
I think the main takeaway from digging into this source is that TUDCA is definitely a real thing, a legitimate biological molecule with known protective effects on cells. It has clear mechanisms.
Right, and the evidence seems strongest, really well established for
those specific cholestatic liver disorders. If that's relevant to you, the answer seems to be, yes, this is a valid treatment. But that implies the prescription form under a doctor's supervision.
Okay. Then there are the areas with, let's say, growing support.
Yeah, that seems to be the metabolic side, particularly for people who are already dealing with NAFL or diagnosed insulin resistance based on those specific studies. The source might lean towards a possibly helpful for these groups, again, keeping those study doses in mind.
And then the other area is direct mitochondrial benefits in humans, neurological support, gut health impacts.
Well, the underlying biology, especially that ER-mito link, is super interesting and definitely warrants more research. But based only on the current human evidence presented in the guide, it's kind of a not yet proven or maybe too early to tell.
And for just generally healthy people looking for optimization.
The benefits just haven't been demonstrated yet, according to the source. The mechanisms are compelling, sure, but the proof in healthy humans isn't there right now.
So, boiling down the practical advice from the source, if you have diagnosed insulin resistance or fatty liver, or maybe reason to suspect high cellular stress, TUDCA might be something to bring up with your clinician, perhaps pointing to the specific study data.
That seems like a reasonable interpretation. But for general wellness in already healthy folks, the current evidence presented here just doesn't support it yet, even if the science behind it is intriguing.
It really is quite a journey, isn't it? Following this molecule from, its natural presence in bile through historical use, diving into these intricate cell mechanisms, and then landing in this nuanced landscape of human research with its specific successes and, well, limitations so far.
Absolutely. There's always that complexity between the lab bench and real-world human health.
Which kind of brings us to a final thought for you, the listener, to maybe chew on. We talked about that really compelling mechanism, right? Linking the ER and mitochondria via those MAMs, and how 2-DCA seems poised to help that connection.
Yeah, theoretically very powerful.
But then we look at the human trials reviewed in our source. Strong results for certain liver conditions, yes. Promising data for specific metabolic issues in certain people. But then less clear results, or even failed trials like ALS for other areas where you might expect it to work based on the mechanism, like neurodegeneration or maybe general mitochondrial boosting in healthy people.
Right. The translation isn't always one-to-one.
Exactly.
Yeah.
So what does this gap, this complexity tell us? What does it suggest about the challenge researchers face, taking these exciting basic science findings, these elegant mechanisms discovered in cells or animals, and translating them into therapies that reliably work for diverse human conditions across different body systems? It really just underscores how intricate and interconnected, yet somehow distinct, everything is inside us.